Hello everybody and welcome back to another Oxen Academy video. Today's video we're going to be talking about one of the most important processes in the human body, transcription, mRNA modification, and a further video, translation. They're all truly interconnected. Transcription, translation, and mRNA modification overall encompass the body's changing of DNA into a protein, and it's done through what is known as a central dogma, the idea that we start off with DNA and then convert that into pre-mRNA and take this pre-mRNA and then convert that to mRNA and finally convert mRNA into our final product of a protein. In today's video, covering transcription and mRNA modification, we're going to be going over the process of DNA getting converted into pre-mRNA in what is specifically known as transcription, and finally pre-mRNA getting turned into mRNA, what happens in mRNA modification. So we can abbreviate this to transcription and mRNA modification. So how exactly does this all work? Well, in order to understand transcription and then mRNA modification, we have to have a good understanding of our genetic information, what is known as DNA or deoxyribonucleic acid. One thing that's important about DNA is that it is double-stranded with two backbones. And within these two backbones are base pairs consisting of four main base pairs, two purines and two pyrimidines, adenine, thymine, cytosine, and guanine. Of course, each of these base pairs have a specific base that they do pair to. Adenine always binds to thymine, cytosine always binds to guanine, barring any outliers or anything of the sort. This is really important because these base pairs give us an idea of transcription and the creation of a duplicate strand that holds the same genetic information when we convert it back into a double-stranded piece of genetic information. For example, since we know one side of DNA, let's just say that we have A, T, G, C, A. We know that the other strand codes for the base pairs T, A, C, G, and T, and thus we can operate between strands of DNA. Additionally, we also know of genetic material known as RNA, which is known as ribonucleic acid, which is single-stranded. And rather than the four base pairs of A, T, C, and G, RNA consists of four base pairs with one major change. Instead of T, RNA utilizes U, also known as uracil, which also binds to adenine. So let's take a look at transcription and the specific process with all of this background information. In our double-stranded DNA, there's a specific enzyme known as RNA polymerase that we will be using in this process. Let's draw out our DNA right here. Draw a portion of the DNA much larger to see and more divided. RNA polymerase in transcription facilitates a larger division of certain pieces of DNA. For example, this piece that we've drawn larger than the others in terms of its spacing would be facilitated by RNA polymerase. This large bubble is what is known as a transcription bubble. Transcription bubble. And it's properly named, given the fact that it is, well, a bubble and it's where transcription takes place. Now let's draw in the base pairs of our DNA, except let's keep them quite vague for now, having only a couple of them in the DNA model that we're showing here. How transcription works, that this RNA polymerase, which in this case let's abbreviate to RP, goes through one of the strands of DNA, which is known as the template strand, and using DNA bases creates an RNA chain of RNA bases. This is ultimately known as a pre-mRNA strand. So let's take an example of how this would look like. Let's just say that this template strand here has a couple of base pairs and a base sequence of A, T, C, G, C, T, A, T, C, G. The RNA sequence for this template strand after RNA polymerase transcripts this piece of genetic information would be U, A, C, G, A, U, A, G, C. Again, remember the fact that instead of T, the thymine base pair, RNA uses uracil. Additionally, an important thing to note is that RNA polymerase has more than one type. There's RNA polymerase 1, 2, and 3. So we can properly name it in this sense. And each of these RNA polymerases, when transcribed, has a different RNA that it ultimately makes. RNA polymerase 1 creates rRNA, RNA polymerase 2 creates mRNA, RNA polymerase 3 creates tRNA. But in this scenario, we're worrying about RNA polymerase 2, which creates mRNA. However, with this pre-mRNA, for example, this strand that we have up here, we can't exactly utilize it to create a protein yet, because there are post-transcriptional modifications that we have to do in order to make sure that the mRNA strand rather, is able to finally create a protein. So let's compare the pre-mRNA strand with an mRNA strand. There are three main steps that has to occur in order to convert pre-mRNA into mRNA. The first one is the addition of a poly-A tail. The poly-A tail essentially adds the base pair A, or rather the base A, to the end of the pre-mRNA strand, and it has one main function as to ensure that the mRNA strand does not prematurely degrade. This is important because if the mRNA strand begins to degrade, either A, it won't be turned into a protein at all, or B, if it does turn into a protein, it would be faulty. The second thing that needs to be added onto the pre-mRNA in order to turn it into an mRNA is a G cap, also known as a guanosine cap. This adds a set of G, guanosine, in a modified form to the beginning of the pre-mRNA strand. And not only does this stop the degradation of the mRNA, but at the same time serves as a place for the ribosome, or a place of conversion between mRNA to a protein, to recognize the pre-mRNA, which is then in mRNA form. So it helps recognize the mRNA. And of course, the last thing is known as exon splicing. Given, for example, a random set of RNA base pairs that we've gotten from transcribing a piece of DNA, we can have a base pair of A, U, T, C, C, or A, U, U, because there is no T in RNA, A, U, U, C, C, A, U, G. Within this mRNA strand, pre-mRNA strand, one might think that this specifically would code for a protein. And while that may be true, there are other variants of this exact strand that can be taken out and remade into a new protein altogether. Exon splicing is the taking out of certain areas of the pre-mRNA molecule in order to form a new genetic code altogether. This new genetic code codes for unique proteins as compared to whether the exon was either spliced in a different area, for example, this area 2 compared to area 1, or if the mRNA was spliced at all. Of course, pre-mRNAs can be spliced in multiple areas, and exon splicing is the scientific community's main theory for how humans, given even in that sense a limited amount of DNA, can have such genetic variation and such protein variation, and is a reason why many scientists believe that even though other animals have much more DNA than humans, they don't have as diverse of protein function or even cell function as humans do. Another important thing to consider when regarding all of these concepts is that post-transcriptional modification happens in eukaryotes. It doesn't happen in bacteria, and it doesn't happen in archaea. Rather, it only happens in eukaryotes and is what has contributed to so much genetic diversity, especially when it comes to exon splicing. Ultimately, we've given a quick rundown on how exactly the DNA is able to go from, well, DNA to a pre-mRNA and then an mRNA. Through transcription of RNA polymerase and the specific RNA polymerase of 1, 2, or 3, and then the creation of a pre-mRNA, which then converts itself into an mRNA through addition of a poly-A tail, a G-cap, and exon splicing, we've finally gotten a product of mRNA. And in the next video, we'll be going over how exactly the body uses mRNA in order to create proteins that sustain our cells.